We're now going to hear from Devin Peterson, who is a distinguished professor of food science and technology at The Ohio State University. He's also the founding director of OSU's Flavor Research and Education Center and faculty director of the Foods for Health Initiative. He's going to tell us about some recent research from his lab and show us the state-of-the-art instrumentation that his group uses to study the chemistry of flavor.
It's hard to talk about flavor without talking about spices. For thousands of years, people have incorporated spices into their diets, not only to enhance flavor, but to also promote health and wellness. If you look at global data, poor dietary patterns are the largest contributor to poor health, more than sexually transmitted diseases, drugs, alcohol, and tobacco combined. Thus, there's a tremendous opportunity to have food do more to improve our quality of life. And one of the major culprits in unhealthy eating patterns is the overconsumption of salt. Salt has a long history of use in foods as a preservative and to enhance flavor. Today, it remains an inexpensive and commonly used ingredient. However, we now know that excessive intake of sodium can pose serious risks to cardiovascular health. We also know that telling people to choose low sodium options really doesn't work. Even when we hear from consumers that they want to eat healthy, what we actually see is that people eat what's convenient, affordable, and palatable. In other words, people eat what they like. We've seen studies from other research groups that adding spices to low-sodium foods can help with acceptance. So we wanted to investigate, is there anything more complex going on than just flavor replacement? Flavor arises from a complex mixture of compounds, specific molecular patterns that trigger millions of receptors in our mouths and noses, collectively shaping our cognitive and perceptual experience. Molecules that bind to these sensory receptors are called agonists, and they activate three distinct sensory systems: olfaction, smell, gustation, taste, and somatosensory, tactile and temperature, which then sends these signals to the brain and we perceive as flavor. For quite some time, my lab has been exploring a unique class of molecules called modulators. These are molecules that don't have a distinct flavor on their own, yet impact flavor perception. Some modulators can suppress perception, whereas others can enhance it. And they typically are active at very low concentrations, at the low part per million range or below. So we set out to see if we could identify specific compounds from spices that would enhance the perception of saltiness when added to food. However, comprehensively extracting compounds from plant materials is technically complex, and achieving purity needed for analytical and sensory evaluation requires considerable time and effort. Dr. Irene Wang, a former student of mine, was the lead on this recent project focusing on identifying spice-derived compounds that modulate the perception of saltiness. So I'll let her explain our methods for identifying and purifying these compounds.
I just finished my Ph.D. earlier this year, and now I work as a flavor scientist at a company called Carry. Salt is one of the 5 tastes that we sense with the receptors on our tongues. And because they are on our tongues, this means that these tastes are most likely affected by non-volatile compounds that stay dissolved in your saliva. So we decided to try liquid chromatography instead of gas chromatography to screen for salt-enhancing molecules. I chose 5 different spices that might affect salt perception based on the literature. Out of these spices, I found cumin and thyme to be promising candidates for increasing perceived saltiness in foods based on sensory screenings. I started by doing extractions. I would dissolve 100 grams of cumin at a time in 1 liter of 20% ethanol solution to leach out all the compounds I could. Then I used multidimensional LC-MS, which is a technique often used for purifying natural compounds, since the starting materials can be very complex. Basically, I would run a column and collect different fractions. Then a panel of taste testers would determine whether the compounds in these samples made a salt solution taste saltier. Then I would take the salt-enhancing fractions and run them again with different solvents to separate the mixture even further. I had to do this 4 times before I got pure isolated compounds. And each time, I did one of these prep columns. I would then have to freeze dry all the fractions multiple times to remove the solvent before reconstituting them in salt solutions for tasting. This is our fractionation room where most of the lab separation work takes place. It's completely quarantined from other chemistry species, and we only use food-grade solvents, so it's safe to ingest the things we isolate. This approach, where we separate, taste, and repeat, is called sensory-guided fractionation. And this process yielded some interesting results. We were able to identify 1 compound in thyme and 2 compounds in cumin that enhance the perception of salt. This was super exciting, since the salt-enhancing compounds we identified in cumin had never been characterized before. We did a full spectral analysis to determine the structure of the compounds, and since we discovered them, we got to name them too. Of the 5 basic tastes, salt taste is undoubtedly the most challenging to study because we have limited understanding of its molecular and cellular mechanisms. Using LC-MS, we are beginning to uncover some of the complexities of saltiness perception.
Irene's work is a great example of how science can be used to enhance the food supply. By having a deeper appreciation for compounds that contribute to flavor, we can optimize food formulations that are healthier yet meet consumer demands. But there's still a lot we don't understand about taste, receptor biochemistry, and all the underlying mechanisms that come into play for tasting salt. So, we are continuing to look for salt-enhancing compounds more broadly and to collaborate with receptor biologists to develop more effective solutions. Generally, when we look at flavor modulators in foods and beverages, we find evidence they exist. For example, we've identified modulators in chili peppers, beer, orange juice, among many others. What we see over and over again is that flavor receptor interactions are complex, and much more complex than just agonist activity. Going forward, we need to take creative approaches to studying flavor perception, which is an exciting challenge. What also excites me is that our work connects to everyday life, asking questions that influence our health and how we experience food. I'm fortunate to have a great group of students, postdocs, and staff at the Flavor Research and Education Center at The Ohio State University. We're a multicultural lab with a multidisciplinary approach working at the intersection of chemistry and human perception. Thank you to ACS for inviting us to help you celebrate National Chemistry Week. Irene and I will be joining you live for questions, so go ahead and submit any you have, and we'll try to answer as many as we can.